Hello, my name is Matthew Evans. I'm an assistant professor in the Department of Microbiology at the Icahn School of Medicine at Mount Sinai.
Hi, I'm Brian Brown. I'm an associate professor in the Department of Genetics and Genomic Sciences at Mount Sinai.
Today, we're here to tell you about our recent discovery, which will be published this week in Nature Communications, about how the hepatitis C virus can develop resistance to microRNA-based targeted therapies. The hepatitis C virus, or HCV for short, is a major human pathogen. It's infected over 3% of the world's population. In the US, that's 3 million people alone. It's responsible for over 2/3 of liver cancer and liver transplants in the Western Hemisphere. In fact, in the US, HCV has recently overcome HIV as a leading cause of disease. Recently, new therapies have become available that can absolutely cure HCV infection in the vast majority of patients. However, these drugs are extremely expensive and thus will not be available to all patients. Furthermore, these drugs also elicit resistance in the virus, which will limit their effectiveness.
And we had some students working on the project, and at first we didn't think we saw much different than what was reported in the clinical trials, but after a while, the students looked, and actually they extended their analysis much longer than had previously been done. And when they did this, they realized that the virus over time was evolving resistance to the therapy. And this was sort of a big eureka moment for us because it had never been reported this happened. And of course, you know, we know that most therapies, at least in the bacterial world, often develop resistance, or the bacteria often develops resistance to the therapy. So it was quite exciting that we had found the same thing for HCV because it was something that could potentially inform the clinical trials. So one of the things that we did when we first made this discovery is we had to sort of figure out what was going on. And a few years ago this would have been something very challenging to do, but now with the new tools in genomics, particularly the ability to sequence a genome very rapidly, we were able to take the HCV viruses which had exhibited this resistance and throw it onto these sequencing machines, and within basically a week we had the entire sequence of the virus. And the amazing thing is we found that there was only one change that was really dominating in the entire viral genome, and this was actually located right near where this gene, the microRNA, binds in the virus.
During the course of our studies, we also found that the interaction between HCV and this microRNA interfered with the ability of this microRNA to perform its normal cellular role of suppressing genes within the host cell. This is interesting because miR-122 actually acts as a tumor suppressor, and the interference by the virus may contribute to the ability of HCV to induce liver cancer.
MicroRNAs were discovered about 15 years ago, at least the bulk of them, and what they do is they actually are an abundant class of small RNAs that regulate almost the entire transcriptome. So they have a major control over gene expression, and in general what they do is they suppress the expression of a gene to control the dose of it. And what was amazing about the HCV discovery is that it was found that instead of suppressing HCV, this one microRNA, miR-122, actually works to stabilize HCV and increase it enhance its infection.
Our findings also have broader implications, not just how this therapy can target the virus, but how microRNAs and RNAs interact in general. If HCV is able to control some cellular growth state by interacting with the microRNA, are other genes also being regulated in this same manner?
So one of the things that made this work possible was really the environment here at Mount Sinai, which really provides a structure for people of very different disciplines to collaborate. So me coming from a genomics background, working with Matt, who's working in microbiology, that's really what made this possible, because we were able to fuse those two disciplines and uncover something that I don't think would have been possible otherwise.
To learn more about our discovery, please read our paper published this week in Nature Communications, or go to our websites to find out more about our ongoing research projects.